Hi, welcome to the Turf Virtual Field Day. My name is Wrennie Wang. I'm a postdoc at Oregon State University. Today I'm going to talk to you about my ongoing research on apicoid endophytes. First, what is apicoid endophyte? Apicoid endophytes are naturally occurring beneficial fungi that form mutualistic relationship with host grasses. In turfgrass, the hosts include fine fescues, perennial ryegrass, and tall fescue.
What I'm showing you here is a picture taken under the microscope. The rectangulars are leaf cells, and the endophyte is stained with a red dye. You can see the unbranched mycelium is growing along the cell and in between the cells, getting the nutrients from the host without causing any symptoms or problems to the host. And actually, in return, endophytes are providing the host with enhanced tolerance to biotic and abiotic stresses. However, those are very strain specific. Some examples. In tall fescue, we see drought tolerance. However, we don't see that in fine fescues. In perennial ryegrass, some endophytes are providing tolerance to insects. And in fine fescues, we see disease resistance is linked to the endophyte. That's where I'm most interested, and I'm going to talk about more. Endophyte has the potential to be used as a biological control, unlike other biological control agents such as mycorrhizal fungi and beneficial bacteria. Those biological agents need to be repeatedly applied to your turf to see the benefits. Once endophytes are established with your turf, they form stable association. Therefore, you do not need to repeatedly apply. I am most interested in disease management. The main concerns with chemical control are overuse of fungicide. Some fungicides are currently working very well for you, may not be working in many years because of the fungicide resistance. And there are increasing restrictions of fungicide use in some areas of our states and neighboring states, and globally in some areas in Canada and Europe. And of course, enhanced tolerance to disease means save us money on fungicides. There are some research in the literature on endophyte-mediated disease resistance. What that means is simply with endophyte you have less disease or no disease, and without the endophyte you have a lot of disease, as shown in this picture on your left. So E plus means with endophyte and E minus means no endophyte. The same turfgrass with endophyte has almost no disease, and without endophyte has lots of disease. Most recently, in hard fescue breeding research, as shown in the picture on your right, they find a hard fescue with the endophyte is very tolerant to dollar spot disease, shown in this row. So the objective of my research is, can we utilize endophytes as alternatives for disease management? Can we improve multiple species of turfgrass with enhanced tolerance to more than one disease?
Very briefly, how I do that. First, I introduce the endophyte to individual grass seedlings, shown here, and grow those seedlings out in the greenhouse, and take some leaf and stain it with a blue dye and look under the microscope and look for this typical apical endophyte mycelium shown next to the arrow. And now those grass plants containing the endophyte will be ready for further testing. In addition to daughter spot disease, as I mentioned earlier, I'm also interested in other diseases. Being in the Pacific Northwest, we have this perfect weather for studying cool weather diseases, such as red thread, leaf spot, and the famous microdocium patch. You typically see microdocium patch on putting green turf, but it can also occur on the higher cut of turf. Our grass plants, with the endophyte introduced, will be tested against different diseases, either in the field or in the growth chamber. I'm very excited about this research and looking forward to getting more results to share with you. With that, thank you for watching.